Hola, buenas tardes. Hello. Well, thank you very much to everyone for attending this online event for the CHIC project, okay? My name is Macarena Sanz. I'm the director of the Spanish consultancy company ID Consortium, and we are a partner in the CHIC project, okay? We are the communication and dissemination coordinator in the CHIC project, and we hope that you enjoy this event this afternoon. I will now pass the word to Mr. Eric Toussaint from CHIC that will be moderating the event during this afternoon. Thank you very much to everyone.
Thank you, Macarena. I can see that it's still light at your place. I'm in Holland and it's very much dark in here. That's a disadvantage of living in the Netherlands during wintertime. Days are shorter than in Spain. Thank you for introducing. So my name is Eric Toussaint. I'm communication advisor at CHIC. That's a research company, a plant research company in the Netherlands. And personally, I'm passionate about plant breeding and about telling the world what plant breeding is so good about. Today, we have an online version of the famous Science Week in Madrid. And thanks to this online version, we now have a lot of people from all over Europe in our panel without the need of they didn't need to travel at all. So that's very nice indeed. Today, I will be your host, and together with Ronald Terpenes, who is not visible right now, we will select questions from the audience to pass on to the panel. So please ask your questions through the chat panel or through Twitter using our @H2020_CHIC, and then Ronald will certainly find your questions. Okay, well, I would like to start with a question to you. Say you got the opportunity to develop a new crop, which is very important for the future of food supply or for agriculture. Could you please fill in the chat, what kind of a crop would you like to develop?
Question?
Right now, I do not see any answers. No bright ideas about crops we need to develop? Ah, okay. Easy to grow and very nutritious. Yeah, okay, very good. A crop to act as fuel, so a biofuel, okay? Thank you, Yara and Jeannot. Okay, well, in CHIC, we develop new breeding techniques, also known as gene editing. And in CHIC, we develop those techniques to improve chicory crops. And we want to develop a chicory crop that produces more food fiber and better food fibers and compounds that can be used to produce medicines. CHIC is a very big team from researchers, communication specialists, artists from all over Europe. And a few of the team we have in our panel. So I would like to give all of them the opportunity to briefly introduce themselves. So Paloma, can I start with you? Paloma Diaz-Perez from Spain.
Okay.
Please introduce yourself.
I'm starting the video. Okay. Do you want me to use the glasses?
Yes, please go on. Please go on.
Well, thank you very much, Eric, for the introduction. My name is Paloma Diaz. I'm a computer science and engineering professor at the University Carlos III de Madrid. And I lead a group that works on interactive technologies, including virtual reality, augmented reality, and these kind of things. Our role in the project is developing educational tools because these kind of things, apart from being cool, can be used to improve our experiences with educational material. So now I'm wearing the HoloLens 2, which is a type of augmented reality device. And now I can see what you are thinking, because augmented reality means that I see the reality, I'm seeing the Zoom screen, but I also seeing what you think right now. And just let me try. I think that 1/3 of you thinks, no way, I'm completely crazy. The other third thinks, I don't know. And the other third is trying to fight with Zoom to make everything working now. So this is basically what we do in the project, and in general in the group. We work on all kinds of technologies, including also mixed reality, social computing, games, applied to many different areas. And one of them, of course, is education, because I work in the university. So I'm quite concerned on how we can use these kind of things to make things easier and to make learning more relevant and more meaningful. And I
Thank
think
you,
that's
Paloma.
all. I don't know if you want me to talk more, and that's
No, you get the
by now.
opportunity to talk more later on, because the last
Thank
half an
you.
hour of our online meeting it will be about gaming and about your work. Thank you, Paloma.
Thank you.
So next is Teresa. Teresa, can I please give you the floor to introduce yourself?
Yes. Can you
Hi.
see? Yes. Hello. Well, I'm Teresa Norati. I work as I'm a professor also at University Carlos III of Madrid. I work with Paloma, so basically I'm going to repeat the same. I have here another classes that I'm going to show you later. And yeah, I work in the possibility to use immersive technologies for navigating big data and information useful for social computing. Thank you very much.
Thank you, Teresa. Thank you very much indeed. So next is Katja, Katarina Tankar from Wageningen. Katja,
Hello.
please go ahead.
Hi. I'm a plant scientist, and I'm also located in the Netherlands, in Wageningen. And my interest is actually this plant that you can see here. This is the chicory, the way we grow it here in the lab. So you see that there are small plants grown in a sterile environment. And these are the plants that we are actually using to do gene editing. And as you said, we are doing two things here in Wageningen. So we are trying to change these plants to make better food fibers and also to make medicinal compounds. So this is what my interest in the CHIC is and my role in the CHIC is, and that's why I can tell you more about this part of the project.
Thank you, Katja. I'm not sure how many of us have seen chicory in real. It looks a bit like a sugar beet, you could say, when you just pass a field and you could say, oh, it's a sugar beet. But in fact, it's something totally different with a total different use. And then we come to Matthew de Rode from Census. You could say a sugar company, but there is more to Census.
I was already nodding my head. No, it's not sugar beets.
Go ahead, please.
But indeed, if you see a field, it looks very much like a sugar beet. That the leaves are almost similar. The root is not. You could say, okay, why are we interested in this root at all? And that is where I come into play. Before a crop ends up on your plate, a lot of things have been done to it. They have been harvested, they have been collected, redusted. If it is a potato, it ends up in your homes directly. In this case, chicory is used to retract food fiber, but also a sugar replacer, so a healthy sugar replacer from it, inulin. And that is what I do. I am the innovation manager at Census, a Dutch company that uses this crop, chicory, to extract a sugar replacer from it. So that is basically my job, and I know very well how the root looks like.
Yeah, you do. Thank you, Matthew. Anna, Anna Dimitriou is artist. She's based in the UK. And in fact, she is together with a partner in crime in the CHIC artist support of the CHIC project. Anna, please go ahead.
Yeah, thanks for inviting me to join this. Talking of chicory plants, I've been growing one. So this is my show and tell kind of thing here. You can see it's in a huge pot because they've got these long roots. And I started growing that after our visit to you, actually, and to Wageningen and in the Netherlands when we came to see all the research that's going on there. And I got given some seeds at the Wageningen plant research. And so this is one of the plants that I've grown from that. And that's kind of what I do as an artist. I really like to try and get into the research and find out as much as I can about what's going on and how things work. So we've been kind of studying this from the scientific perspective, but also from a kind of cultural history perspective. And I can talk more about that a bit later.
Okay, thank you. Well, it was fun to have you in our lab, because you really were in our labs, and also at Wageningen University Research, you also were visiting the lab and really participating. And it was really fun to have you there. We have learned a lot from that as well. So, and of course, an artist has a very wide audience. And that's where we go to Armin Spök. Armin is, I could say, I think, social scientist. Armin, can you introduce yourself?
Certainly I can. Hello, everybody. My name is Armin Spök. I'm a science and society researcher, and explain to you in a minute what this is about, at the Graz University of Technology, based in Austria. So the type of work that I'm doing is probably something that very few people are familiar with. So I'm just in between the science and the social science. Effectively, I started off as a molecular biologist, but after 20 years now, I've arrived into social science, and I'm particularly interested in the relation between innovation, novel technologies, and societal issues, societal constraints, societal demands, and also in the case of GMOs, of genetically modified organisms, also rejections of at least parts of the society to these novel technologies. That's my particular interest. So as I mentioned, I'm now a social science researcher, so that means that I don't have any stakes in these plants. It's just for us of a huge interest to understand what are the reasons why people would like to adopt novel technologies and why they are perhaps rejecting them. So we are interested to identify those factors. And that was also the reason why I got very much interested into the CHIC project, because CHIC is employing a quite very interesting new technologies, new breeding technologies that are, as we see it, quite different from traditional genetically modified crops. And our main interest now is to understand how stakeholders and broader publics perceive these novel technologies, these novel breeding technologies, in particular if they're used on chicory and root chick to develop a novel type of chicory. And what are possibly hindering and facilitating factors for them. So, and as a second step, we would like to feed what we learned from stakeholders back to the innovators in our project, so that they can try to understand how stakeholders think, what their concerns, what they would like to have, and try to adapt to those concerns and demands.
Okay, thank you, Armin. Thank you very much. So thank you for introducing yourself. I think I've got everybody, right? And let me start with a question about this crop, because in the end we're talking about a plant, about a crop. Matthew, how many square hectares, I should say. How many hectares are being grown from this chicory crop in Europe, do you think?
In Europe, about 15,000 hectares, and then mainly in the north of Europe, so the northwest, so the north of France, Belgium, the Netherlands, and a little bit of Germany. And
to give us a little bit of context, 15,000 hectares. How many hectares of sugar beet do we have?
60,000.
60,000. So that's
much
almost
more.
A quarter. But
And
it's
then,
almost a quarter.
Yeah, but I must say that chicory, we use it to make that sugar replacer. But in the north of France, an important part is also made for chicory coffee, so the coffee surrogate that you use. And there's also a large part that is made for, that is grown for the cicoria, so for the salad, the white leaves that you can eat. So that's also part of that acreage.
As a real vegetable?
Yep.
Okay.
And then you eat the leaves.
Okay. And you said something specific about those products. You said it is healthy. It's a healthy sugar.
Indeed.
I guess not all of our audience knows why these compounds are healthy. Can you explain briefly about it?
Yes, I can. I mean, it is the fun part is that this inulin, this healthy sugar. Formally, we cannot call it a sugar, but let's do it for convenience. So this healthy fiber, we cannot digest it. So that's a bit odd. So what we do, but the fun part is that it comes then completely unharmed in the gut. And the gut microbiota, so all the bacteria that are present in the gut, and there are millions of them, they consume this healthy fiber. And by having a healthy gut microbiota, you become, well, basically you can say you become a healthy person. If you have a healthy gut bacteria, you are a healthy person. I mean, there are a lot of links between a healthy gut bacteria and a healthy person. So that link is almost direct. So therefore, this ingredient contributes to, hey, my dinner is being brought down here. So, sorry. No worries. If you hear a lot of noise, then it is me eating.
We'll shut you off. No worries.
So this healthy fiber is helping, contributing to your health, and the extra benefit is that it's also slightly sweet, and therefore you can even replace sugar for a great deal with it. So it is in that way, it is a healthy sugar because it's also sweet, and your gut microbiota thrives on it.
Okay. So that's about, sorry.
So, and just to give you a bit of it, if you just say, well, how do we discover this? Can I not eat it? Can I not eat the root itself? Well, the root itself is very bitter, and that's another part. But it's also inulin is also present in garlic, is also present in onions, is also present in leek. So if you eat onions, if you eat garlic, you also get your doses of inulin in your daily doses. So you already eat it in normal and the only thing we do is extract it and add it to other food stuff.
So Armin has an inspiring question. What's on your dish? Is there any chicory on your dish right now?
No, there's a very typical Dutch Indonesian dish is satay.
But there should be vegetables in it.
I have spinach here.
Okay. That makes sense. So that's about the plant.
Yeah.
And of course, CHIC is about plants and it's about technology. Katja, so when I think of the process of making a gene edit in this chicory plant, when I work in the lab, what would it look like and how much would it take? I get a plant from Matthew, from one of his fields, and I want to conduct gene editing on his plant. What should I do? Briefly, of course, because I know it takes a long time, but briefly explain. What do we see? What do I do? And how do I end up with a plant with an edited gene in it?
Yeah, I have a few items to show you there. So I already showed you that the plants that we have, that we grow here, they are grown in vitro. That means that there is a nutrition added into the agar, and the plants can grow in sterile condition. And what we do with these plants is that we take one such small leaves, a fresh leaf, and we isolate single cells from those leaves. And in there, we can bring in the Cas protein and a guide RNA, and those two elements are needed to change a few letters in the DNA of the plant. And what happens then is that these single cells, they start growing. I'm showing them to you now.
Yeah.
So the single cells, they start growing into clumps of cells, and all of them are having those letters in the DNA changed that we have made in a single cell. And what happens in the next phase, so these ones have been prepared in September. So you see that they are already quite large now, 2 months later. We have from one cell, many cells. And this is a bit difficult to see, but I hope that you see that the first ones are already making small leaves. So there are first plants being formed from these single cells.
Mm-hmm.
And from these cells, we again regenerate plants. So we get another plant, and later again, they will look like that. In 3 more months from now, they will look like this. And what we do then is we look at the food fibers, and we look at the terpenes in those plants and see whether the change in the DNA has also resulted in a change in the profile of those compounds in the plant.
Because in the end, that is what it is.
That is what we are trying to do.
So
these are not random changes, of course. We know exactly. What we have also studied is the genes in these chicory plants, and we know which genes are responsible to make the food fibers and which genes are responsible to make the terpenes. And in those genes, we bring in a little change. And that's how we make a plant. It takes a few months actually to get a gene-edited plant.
And those genes are involved in the biosynthesis. So they are generally coding for an enzyme or something like that?
Yeah, these genes are involved in the biosynthesis, or they are involved in the degradation of those compounds.
You want to stop degradation and you want to
make more healthy compounds.
Okay. And so you know a bit about the healthy compounds. We are also talking about substances that may be used for medicals, for drug production. Do we know so much about chicory now that we can think of any kind of trait and just look for the gene and then make an intended change in the DNA using gene editing and be pretty much sure that you really alter the right gene?
Now, for some genes, let's say when we talk about healthy compounds, besides inulin, for the inulin biosynthesis pathways, all the genes have been discovered already. But chicory also makes terpenes. And these are small molecules that have health properties. And for those, we know how the biosynthesis starts, but there are several steps in the biosynthesis we are still discovering. So in that case, we maybe have 5 gene candidates, and then we try which of the 5 will make the change that we are looking for.
And do you know any terpenes we may know from?
Yeah, actually, terpenes are a very large group of molecules.
Okay.
Plants make 40,000 different terpenes.
Oh, really?
And plants use these terpenes to communicate to their environment. So to attract the pollinator, or to attract us to eat a fruit, or to defend themselves. And many famous terpenes are already used by us, like menthol that you have in your toothpaste every morning. Or
so you can even smell them.
Yeah, you can. They're very fragrant. They're very tasty. Citrus flavors, those are also terpenes. But there are also terpenes that are famous because they are medicinal. And those you can find in the cannabis plant. Those you can find in the taxus plant. So the taxol, that is an anti-cancer molecule.
It's also a terpene?
It's also a terpene.
Oh, okay.
And I think another one that has been very famous the last few years is Artemisinin. and that is a very potent anti-malaria medicine. And actually the chicory terpenes, when you look at the structure, they look very much like artemisinin, like this anti-malarial terpene.
Okay. And I see that Matthew commented, yeah, but those terpenes in chicory, they are not so tasty. So, yeah. Well, you want to use them for something different, right? Not for say a flavor or something like that,
no.
Right?
The chicory terpenes are indeed very bitter. So when you isolate the food fiber from chicory, you actually remove them so that you retain the sweet taste. And what we are looking at in the CHIC project is whether these terpenes have medicinal properties, and they can be used then as painkillers. They can be used as antibacterial or antifungal compounds, or they can be used as anti-inflammatory compounds. And in the CHIC project, we are actually have a group of laboratories that is researching their properties, and a group of scientists that is then changing the terpenes in the chicory plants.
Okay, okay. We can see you're working, and you're at your working place.
Yeah.
There's a lady cleaning.
Yes,
the
there's
right
yeah.
No worries, no worries, no worries. So we got a question from the audience. There is no name to it now, I think. I think it's a question for you, Katja, or you're a good person to ask this question for. So when you know this technique well, and you know about a lot of plants, how well we know the genes and where they code for, would it also be attractive to use gene editing in vegetables? And could you give an example?
Yes, I think you can use, it is already applied in many vegetables, and especially for the plants that are used as crops that are very well characterized, such as major crops, maize and soybean, etc. The genome is also very well known. So there it is very easy to apply these techniques.
Yeah.
It's also applied in vegetable crops such as tomato and potato, and it's less applied to minor crops such as chicory until now, or to medicinal plants, because those we don't know so well yet. So we don't know all the genes yet. And well, especially for those plants, there is still a lot of research that needs to be done to know how they make the healthy compounds, but also to be able to use gene editing in those plants.
Some kind of orphan biosynthesis traject or something like that. Yeah. Nice. Okay, well, thank you for explaining about techniques. And well, we started with Matthew talking about the plants. And well, Anna is faced with all of these for probably the first time in her life. Anna, wasn't it? It was the first time in your life you saw a chicory plant when you came to Matthew?
I think so. Yeah.
So
they sent them through the post festival. They sent some roots to us.
They did. Yeah.
Scanning. So yeah.
So you're faced with a new plant about which you didn't know any background, what it was related to or whatever, and a technique you may have heard about, CRISPR perhaps. Because CRISPR has been in the news even some years ago already. And this comes to you, and then you can do, as an artist, you can get inspired by those things. Can you explain us a little bit, in general, when you're collaborating with scientists, how does it work that there is an interaction between you and the artist, between you and the scientists? And how do you extract things that you can use for art?
Sure. Well, I've got some images that I could show of my visit to you. So that's probably the best way of explaining. If I can share my screen and show you some images.
Yeah, we are seeing your screen in PowerPoint.
Yeah, good. And now just get there. Can you see that? So
yeah, nicely.
So I'd actually done some work with CRISPR before. I'd done CRISPR modification artwork. So I work in a field called bioart. So I work kind of at the cutting edge of new technologies and also the history of science, and particularly the history of microbiology. But I hadn't really done any work with plants before. So I was very excited to do this. And what's been fascinating is like the experience of coming to see the labs and the research and understanding the differences and the similarities. So this is a sort of very grand and luscious chicory plant growing in the greenhouses at Wageningen Plant Research at the university there that I came to see. And this is the box of chicory roots that we got sent in the post from Matthew at Census during the first harvest. And we were kind of rummaging around in this huge box and doing some of the techniques. So we kind of did a DIY staining technique. So this is a cut through of the roots, obviously working with Alex May that you mentioned as well. So we cut through the roots and we stained this with, I used textile dyes to bring out these kind of highlights. And I think these are the terpenes, if I understand it, in some of the feathery shapes here that you can see.
And it was a total surprise, of course. The picture you get is a total surprise, right?
Yeah, kind of, yeah.
Okay.
And then I tried natural dyeing with chicory. So I got some pigment. It has some pigment. I think if I mordant the fibers more. So I kind of playing with the medium and the materiality and then thinking around the concepts of it. So things that I was really inspired by are these two books. So there's Heinrich von Offerdingen by Novalis, which is the kind of central book for German Romanticism, the Romantic movement, which was a kind of antithesis of the Industrial Revolution. So they were kind of against, so nature was on one side and industry was on another side.
Yep.
And this is this very romantic book. The central form in the book is the blue flower, which many people think he's referring to the chicory plant. So it becomes this symbol of German Romanticism. So you have this beauty, because it has this beautiful blue flower, it grows wild on the roadsides, and there's a lot of mythology around chicory. And that book by Novalis went and inspired Goethe, the famous scientist and artist, whose Metamorphoses of Plants went on to inspire Darwin. He mentions it in Origin of Species, who had this idea of that there was some sort of primal or original plant. And he talked about the blue flower being like the inspiration for this. So I was thinking about these people, and we were getting into this like thing of what is the plant, what is the root, what is this mythical plant? But the root's more interesting than the flower in sort of scientific terms. So we 3D scanned the roots. These are some of the 3D scans. This is a kind of journey inside the 3D scan of the roots. So you're sort of traveling down. It's kind of like in a space of the plant, really. Seeing it from a side, you could never normally see it. And
this is already a picture I would like to have in my house.
And these are based on 3D scans of the leaves. So we scanned the leaves and created these kind of wireframes of it, which you can fly through. And we're working with 3D printing of the leaves and the plants and things. Also, what Katja was talking about, I was able to go and visit the labs and see these are the leaves being sort of impregnated with the agarose with the agrobacterium to put the CRISPR edit in there. That's a very long process. I haven't been able to do that yet, but then I visited Keygene, where you use a different technique with the working with the protoplast to do the CRISPR gene editing. So I was able to do that there, and these are some of the images of the protoplast that I was able to go and work hands-on in the lab and modify with your colleagues. And there's some of the plants, the little plants that we saw growing there. And what I was so shocked and kind of impressed by, and it goes back to Goethe, because he talks about the idea that a plant could be created from the leaf of the plant. That's the beginning of it. Yeah, he mentions that in Metamorphoses of Plants. And when you use these sort of growth factors in the agar for the plants, you can kind of make a plant grow out of just either a cell of a plant or a bit of a leaf. And I didn't know that, so that was really strange. And to see that he talked about that, and this is some of the CRISPR edited protoplasts that I was able to do, and I'm really looking forward to coming and
so protoplasts are in fact plant cells without a cell wall, right?
Yeah, and then
that's why they are football shaped. That's why they are football shaped.
These have got their CRISPR modification in to knock out the terpene gene, the bitter terpene gene. So I wanted to kind of really embed myself and learn about it. And then things like in the future, I want to kind of grow in using you've got a wonderful 3D bioprinter at Keygene, and I've really got ambitions to kind of work with that as well, and kind of grow kind of sculptural shapes with the edited protoplast growing out of it and things based on our scans. And then I was able to go, Alex and I went and visited Matthew at Census, and he was like, don't take a photo of this. This is just some weird metal flaps that we have to stop the splashes, but from an artistic perspective they're just beautiful. And so I was like, oh, that's lovely, I'm going to take a photo of that. He's like, don't take a photo. But it's beautiful, and people see it and they say, what's that beautiful photo? So there's you can see the beauty in the industrial settings and things, and that's us going to visit Census there. So the big thing I think for me is this sort of really embedding myself into the research, but also into the history. And the central question for us is something that we discussed with you when we visited, is this idea of what is natural, what is nature? Because all the plants and foods the plants that we eat have all been through loads and loads of breeding techniques, and this technique that you're using kind of throws that into question again. And that's where it ties into this idea of the Romantic period and it being against the sort of separate to the Industrial Revolution. But now the chicory plant is here central to this new biotechnological revolution using CRISPR. So it's in a much more complex position. And that's what I'm interested with Alex in kind of exploring now in the next steps.
So I know that scientists are involved in research projects in the European Union, but to a lot of people, this may be a bit of a surprise, Anna. How does a scientist get involved in communication? Artists get involved in communication about science. Do you need to have a diploma for it, or the network, or how do you get it?
It's just through fascination and being kind of really interested in this stuff. I've worked with a lot of scientists over the years and really go in depth into the science. And now it's sort of reached a stage where my kind of in-depth knowledge, which was very much focused, I guess, around two sides of like artificial intelligence and also bacteria, microbiology and infectious disease is a big interest of mine. And then CRISPR and synthetic biology research I started to do tied into that. And then started to come and move on. And now I've started to start exploring other organisms and looking at plants. And what's really interesting is how similar they all are and things. And I've started doing some work with some scientists working with stem cells and epigenetics now. And that's kind of really interesting because it's explaining back to me some of the stuff that I've learned with the chicory work or with the bacteria, and I'm like, oh, well, that's another bit. So I think as an artist, I have this kind of sort of weird patchy knowledge, but very in depth in parts, and it's done in a very hands-on way. So I think the way of like doing it is just to kind of to do it really, and to meet more people, and then there's introductions are made, and then other things happen. And quite a lot of the time, I'm also approached by the scientists as much as I am approaching scientists. So it's become this kind of two-way thing now.
Oh, thank you, Anna.
Thank you.
I've got a question for Matthew, because, well, we've seen the technology, we've seen the plant, and we've seen how it inspires Anna and other scientists. How does it inspire you, Matthew, in thinking of where could gene editing be useful in my business or for my clients or for society? I know that, of course, scientists that develop the technology, they are very proud of what they develop. And you're on the, you could say, the market side. There's the demand for innovation. So for what kind of applications or for what reasons would you, coming from industry, being involved in sugar beets and other plants, when would you think of gene editing as a tool to improve the crops?
I never think of gene editing.
Oh, okay.
No, but first of all, that curiosity that you have as a scientist, that is not different in industry.
No, of course.
I mean, there scientists are working in industry as well. Only
sure.
we work with a different emphasis. We want to please the customer with the best products, and therefore we make money. So you always are curious what is happening around the world, which I could learn from, and then could build my industry further than we do.
Sure.
I mean, our company, the bigger company, exists for 125 years. Now, you do not exist for 125 years if you keep doing the same stuff over and over again. So you have to innovate. And therefore, scientists are working in the industry to make contact with other scientists that are working in academia, with artists like Anna, to get inspired and, hey, this might be a good idea. And for this specific one is exactly the first question from Yara, or the first answer from Yara when you said, what crops do you would like to have developed? And see, I think that Yara, if it is not a free mode, then please correct me, easy to grow and very nutritious.
Yeah.
And this might give an answer exactly to, because we have a crop that has it has 17% of inulin, which we want to have. Now, but if it gets colder, then the plant starts breaking down the inulin. If this research finds out, hey, we can stop that breakdown, or we can make even 20%, 25%, this is completely ridiculous.
Yeah.
We can make, so in fact, the scientists are trying to find out if they can make a more nutritious crop. So exactly the answer that Yara has given here. What would you like for a crop? A more nutritious crop. So we have the crop, and here they are looking for improvements on that crop, that it can be even more nutritious. An extra, which is now that they are also looking for, hey, but this crop also contains very interesting components that might have another health benefit, even against cancer. Whoa, that's something different. And you're not making food. You can even make medicine by growing your plant, as you always have done already. So that is for me so what is the reason for me participating and looking very carefully at the scientists, what they develop? Is very important for our company.
Yeah. Hey, Armin, and in your work, of course, it's important to keep an eye on all stakeholders, also industry. What is your perception of how the different stakeholders in general, right? Because you cannot give all details for every stakeholder, but talking about these tools, plant breeding, new plant breeding techniques, can you say anything in general what stakeholders like industry think of when they hear about gene editing?
Well, I wouldn't be able to say something about stakeholders' views in general. With industry, it's more easy, at least as it's about, as long as we are limited to the plant breeding industry and the biotechnology industry. And well, those guys are very much excited about these new techniques. In particular, as they consider these new techniques as a major improvement compared to genetically modified organisms what we had so far. So it's much faster, it's cheaper, and it's more precise. So therefore, they're quite excited and they are eager to develop products which can be on the market. On the other side of the story is, at least in some countries of Europe, the consumers and stakeholders are kind of reluctant to go for genetically modified crops, and for the time being, even with these genome edited plants, they are putting them into the same basket.
Oh, really?
So that's and there's a reason behind why they're in the same basket, is that according to a ruling of the European Court of Justice, which you see, these newly developed genome edited crops have to be regulated by the same piece of legislation. which many plant scientists and experts think, okay, wood is not totally appropriate. But that's say as things are for the time being. And if you look at the European context, the views of other stakeholders than industry, they're quite different. In Austria, where I'm based, the consumers are very much reluctant to go for these new techniques, but there are other countries in Europe, that, for example, you can consider the Netherlands or UK, where consumers and stakeholders are much more interested in that. So I would say I would imagine that we will see quite a different views all over Europe, with some countries more eager to adopt this and to use these new techniques.
Okay, thank you. So we got another question for Anna. It's a question from Simon Hall. Thank you for your excellent talk. What's your plan for the final piece with the 3D printed sculptures? Or how do you plan to exhibit your artwork? And where can we see it and maybe even interact with the things that you make?
So, I mean, we've still got a while to go, so there'll be bits added to this, but the sort of central concept at the moment is that it will be a full chicory scanned plant, where it is 3D printed, and that the leaves and the root and all these elements of it are somewhere between natural and kind of digital. So they're sort of like the wire framed kind of shapes that you could see in the leaves in the image that I showed, these sort of textures that you could see in it. So it'll be a combination of those, and then the more natural forms, and then this will be in a plant, and then a kind of on a plinth, and then somehow inside that will be the CRISPR modified protoplasts or leaves, the relics of the leaves, held in this kind of sculptural form. So I've got some things like these that I've been working with, and these are the CRISPR, these are some of the CRISPR modified chicory leaves that have been sterilized before they left the lab, and then they've been pressed in a flower press that I've been working with, and then these are ordinary chicory leaves, chicory flowers unmodified. So there'll be images like that. And then there's also this idea of the chicory genome, which is this huge genome with loads and loads of repeating parts of it. So this instruction book for building chicory, I guess, the DNA, the genome of the organism, and it has lots and lots of repeating sections in it. But Goethe, this historic figure of science and art, talked about this idea of the primal plant, and maybe the primal chicory plant was one without all the repeating sequences. So the repeating sequences appear through kind of its evolution, and so we're interested in using the actual genome that and the project's developing a new genome for chicory, a new better genome for chicory at the moment, but we have an early version to work with, and this will be this will be like edited so that these repeating sections aren't in it. So you'll be kind of enveloped by maybe a huge kind of installation of the genome as well, surrounding this plant. And in terms of where we'll show it, we're going to show it a lot. We want to show it in lots of places. We've already got some shows lined up. We're in talks with a museum in the United States as well to show a version of the work. So yeah, but we're definitely looking forward to showing it in the Netherlands and beyond. Okay, thank you. Even in Corona times. I've got quite a few shows on at the moment, actually. Okay, yeah, I've got at least one show on in the Netherlands now. Okay. Shut for a few weeks, but. So, thank you. Katja, there is a question from Charlie. Maybe I should pronounce it slightly different. Charlie Karsel. And I think it could be a good idea when the two of you have contact together, because Charlie is doing a PhD on Artemisia annua, the crop you know, for the production of this anti-malaria drug. And he's very much interested in what types of terpenes
do chicories make, and what's the biosynthesis? Perhaps it's good to tell Charlie that he can find your contact. He can contact you through the website of your employer, Wageningen University Research.
Yeah, that's true. So I can also send it in the chat.
Yeah, please do.
Now.
Please do.
Yeah.
Please do. Thank you. Thank you very much indeed. So we've heard some things about this gene editing, and now I would like to get back to our audience again. Maybe you've also studied a little on gene editing and maybe how it works and what you can do with it. When you think of gene editing, and say you would be a scientist at Keygen, and you will get money to do research, but using gene editing, is there a specific kind of innovation that you would like to develop using gene editing? So intended small alterations in the DNA to develop something that is maybe valuable in earning money or helping the planet go around? So audience, please, could you give an example of what you would like to develop when you would get a lot of money from me to do research at Keygen? I'll give it a sec. I guess Charlie would like to develop a plant that produces more Artemisinin. Okay. I see that Charlie has indeed received an answer. Thank you, Katja. So to make the step to slowly the step to our computer friends, computer game friends, when they introduced themselves, they were very enthusiastic about what they do. And of course, we are all very much enthusiastic about what we do. And I would briefly like to hear from Armin and maybe also from others. Why is it exciting to be involved in this research? It's chicory and it's technology. So Armin, when I could start with you, please. Why is it exciting to be part of this CHIC program, working on a plant, chicory, and techniques like CRISPR?
Well, there's not just one answer to your question. I think, as I mentioned already, I started off as a molecular biologist 20 years ago. So I'm since then I'm kind of fascinated what can be done with molecular genetics. And when I then continued research on the science, technology, and society relations, I found out that due to the pace of the science, there is always something new. There are always new opportunities, but also new challenges and new controversial topics coming up that are being perceived differently by some stakeholders, and the question how to deal with the technology, what kind of how to regulate it. Are there any risk associated with the technology? And if so, how are we going to deal with these particular risks? What particular public support should be given to certain types of innovations? So all these questions are coming up again and again. And with genome editing, this is also and with CRISPR-Cas, this has made now a big wave in the debate and now in the life science debate. So that's probably why I'm quite interested to and quite happy to be part of the CHIC project, because this is essentially at the heart of the public discussion right now.
Yeah. And it's super hot now. The CRISPR inventors, you could say, got the Nobel Prize for chemistry. Was it? Was it for chemistry, Armin?
I think so.
Yeah.
But please
so
correct me.
That gives a lot of course, of exposure to the technique and to maybe also to the perspectives of the technique, both in human medicine but also for plants, I guess. Okay, thank you, Armin. And Matthew, how about you? Why are you? I know you are passionate about your work and about what your company can do for the world. Why are you specifically excited about being part of the CHIC program?
Basically,
or are you excited?
No, not so much. Couldn't care less. No, but basically the broadness. Normally, if you see only for the other viewers, you see only a small part of the participants. I mean, we are with 17, let's say, different participants. Normally, that is a chaos. And in this project, I mean, you don't know that up front, but in this project, everybody collaborates together. It is a very great group, and also the broadness. So yes, we are looking at the techniques.
Yeah.
But more importantly, I think that's the most important part of the project. We're also looking, and we know that these new techniques, like Armin already explained, that these techniques, they are questioned by the people, the European people in particular. And this project emphasizes also on that aspect. I mean, what do you think of this technique? I mean, we don't say the fact that we can do it does not. Well, yeah, we would like to know what you think about it. And Anna is playing and the other artists as well are playing a very important role in that communication. Look, this is important. What do you think about it? And also getting to know the crop. Getting to know the root that's normally under the ground. She gets it out of the ground and makes it a whole new 3D experience instead of a root that is normally in the ground.
It's just also fun. It's also fun, right?
What is not to like about this project? Even if you're not interested in inulin, in chicory or terpenes, it's just fun to be in this project.
Okay. Thank you. So we gradually go to our computer department, you could say. Paloma, could I give you the floor? And the next minutes or several minutes to say a quarter of an hour or half an hour, we'll see how it runs. The floor is yours. In the meantime, we will keep an eye on questions, and when we can, we will answer them also in the Q&A. And we will, when there's time left, we will also answer questions, through the video when we can. Okay, Paloma, go ahead, please.
Perfect. Thank you very much. So we're last but not least, we are the computer scientists here. As I said at the beginning, we work on different kinds of technologies, and our main contribution to the project is trying to help people to understand what these techniques are. Why they are good for. So we were part of the project to be able to address some learning needs for students from Key 7 to Key 12, that in Spain would be the end of the secondary school and the last 2 years of the baccalaureate. Sorry. And what we did was trying to use technologies that can be can act like a hook, because they are very interesting, they are very novel, and everybody wants to use it. That's why we decided to go for the augmented reality glasses, because we had the possibility of creating a game that you can play everywhere. You can play it in your room, you can play it with your family. And there, I will put a video right now so you can see how the game is is going on till now. And the idea is that you augment the reality and you create your own crop, you take care of it, you grow it, and at some point you decide that maybe why not using these techniques to produce something that is healthier or even that can be used for other purposes. And this is when the gene editing part starts. So let me see if I can share the I cannot see where the video is. Oh, I think it's here. Can you see it? No. Well, I have problems with Zoom. I don't know why. Just 1 second to see. Okay. Can you see it or not? Nope. Okay, Teresa, can you please share the video?
Yes.
I have some problems with Zoom. Okay, here it is. Okay. Yes, now we can see it. So you can start. So this is the video we did for Augmented Reality. What we would have liked if we were not in the pandemic time would have been to bring the glasses and to experience it, but we can't do that now for obvious reasons. We did in March. It was the last presentation we did in Madrid. But if any school is interested, we can, once things go better, that at some point they will be better, we can do this kind of on-site experiences. So this is the first game we are going to see. This one has to be played with the HoloLens. HoloLens is this kind of glasses we have here. They are quite complex, and they make it possible to augment reality and to interact in a almost smooth way with reality. So what will happen is that when you wear the glasses, you can put the seeds in the space you are, you can grow the plant, and you see a number of things. So Teresa, please run the video so they can see the game.
I don't think that you can hear it.
Yeah, a bit.
A bit.
So the idea is that, as you can see, Telmo, who's playing, he's in the lab and he can see the reality. And then when he starts wearing the glasses, new things happen, and he can see these augmented things. So he's buying probably seeds. And it runs as any game. You have money, you have to buy things, and you have to take decisions. This is part of the educational motivation, because you have to be careful of the things you do, because you can run out of money without having caught any specific goal. So once you have the land and you have the seeds, the chicory is starting to grow. As you can see, so you can have different plants and you can take decisions on them. Also, some situations might happen with the plant, because you can see how healthy they are, how they are growing, and which is the price in the market, which is very important, because if you sell them, then you get money. And the idea is to get the more money possible.
I like the checkout sound.
Yeah, very commercial. Also, our plants have some weather conditions. You saw the storm, but also the sun, and you have to react to that. And then we have these insects, which is a fly. You can't hear it, but when you are wearing the glasses, there is this noise.
Oh, yeah, terrifying.
And this is really horrible. You have to kill the flies, not because they are going to kill the chicory, because they are going to killing you with a headache. So now Termo is trying to kill the flies, using his hands. As you see, this is one of the things you can do. We don't have yet the lab part here because there are things that have to be changed in the lab concerning how the DNA chain is changed. But the second part, which is not in the video, will be how to work in the lab to take one of these DNA chains and do some specific changes, as Katrina was saying before, and then see what happens when you do this modification, which, as she said, is not random, and that can produce more money and still win. So this is one of the options we did, which is for augmented reality. In the other case, we were experimenting with virtual reality, which is something completely different. Now, Teresa will probably show you how the glasses look like. But the difference between augmented reality and virtual reality is that in virtual reality, you disappear from this world. You move to a new world, which is the virtual world. So you don't see our world, you don't hear our world. And this is very good in terms of education because you get immersed into a new activity. And if the activity is engaging enough, you forget about time. We all know this, time flies while you are having fun. Mm-hmm. But this is one of the things that happens with virtual reality. You go there, you start moving and you start doing things and you forget about things. It's the same like in games. I think that our students understand that very well because they have the same flow experience in the games. So in this case, what we have could be called we call it the bubbles, the CHIC bubbles, but it could be like a Chicpedia because it's like a Wikipedia about terms related with gene editing and chicory. And it is created using semantic bubbles that are organized as a hierarchy of terms. And the goal of this game is more learning about the concept, exploring the concept from different perspectives and navigating through one space to get more information. It's kind of less fun than the other game because in the other game you are doing things, you are earning money, you are playing, basically.
There's a challenge.
Yeah. But in this case, what you have is to explore the space. And since the space is quite big, a good way to explore the space is using virtual reality. And now, Teresa, I think I will give you the floor so you can explain how the game works. And I think they can also try it because you will share the URL. If you have a computer, you can connect. There are very few directions on how to move on this space, and you can use it also in the computer. You don't need the glasses. Teresa will use the glasses because we are kind of computer nerds, but you can use it also in the computer. So please, Teresa, explain the game.
Thank you very much, Paloma. I'm trying to look for the chat. I'm going to paste the link in the chat so people that want to use it can. So the idea of this game is to use it with a glasses like the one that I'm wearing. Okay, so the particular thing of these glasses is that they are not like the HoloLens that Paloma were showing you. They don't need to be connected to any computer. So they have no cable, so you can just use it, wear it, and start using it. They have these controllers, and they are quite used to interact with. Well, for presenting the bubble and just the application, I'm just using my desktop. So when you open the website with the visualization, with the virtual space, you are going to have, first of all, you are going to see a description of the instruction for using for exploring the space, both with a computer, a laptop, or with a glasses. So we explain both how to use them in both cases. And as Paloma explained, this is a sort of encyclopedia that you can explore for learning more about the topic we are discussing here. So we also propose here a sort of learning activity that could be used, for example, in a school or during a fair or in a education, it's like an education activity that people can use. The idea is to explore the bubble, trying to learn more or learn the most important concept about the CHIC project and the genetic edition in agriculture. So this is an example of activity, but you could build your own learning activity. So the idea here is to present a sort of questions that people have to answer to this question, exploring the space, exploring the bubble. So for example, we have what techniques is used for genetic editing in agriculture, or where do you find inulin, and so on. Okay, now we are going to see how to answer to this question. So first of all, I'm going to show you the space. When you open the space with your glasses, imagine that you are wearing a glasses, and what you see in 2 dimensions here is all around you. So you start looking around you and you see the entire space, and you see all the this bubble, all this sphere around you. As you can see, each one of these spheres is labeled with one of the journalistic questions, because our idea of structuring the knowledge in this case is using a journalist approach. So we look for answer for when, why, what, how, where, and in particular, so this organization can be applied to different topics. In particular, here we apply this organization to the CHIC topics. Okay, so you can see that all the spheres are like transparent, so you can imagine that inside there is something. For example, inside how, I can see that there are a couple of spheres more, or inside what, I can see at least 5, 6 spheres. So if you enter, now you can enter inside one of them, and you can see what you have inside. For example, try to, going back to the questions we had, trying to answer to this question, where do you find inulin? I underline these words in order to suggest how to reach the answer to this question. So in this case, you have to enter inside the where word, because the where category, because the question is about is a where question. So we reach the bubble, we move in the space, we reach the bubble. Of course, the navigation as well as the interaction is smoothly in using the glasses. Okay, it would be a little bit difficult using the desktop, basically because they've been designed for an immersive glasses, virtual reality glasses. So now we are inside the where bubble. We also have a menu, clicking one of the keyboard, where we can see a sort of breadcrumb, or we can see where we are actually. And we found here, for example, the word inulin. So if we want to know where the inulin is, or this component, where we can find this component, we can enter and find chicory. That is basically the answer we are looking for. Once we reach the final bubble, to call it in a way, we can still enter in it. We know that there is no other bubble, because in this case we cannot see other bubble inside. But if we enter, we can see that we find some multimedia content that we can that give us an idea about this concept. For example, in this case, we find a text, a description of the chicory concept. We also find a video explaining what chicory is, and we also find a 3D model of the chicory plant. So the idea is to put inside each one of the bubbles this multimedia content that give us more information about the concept. It's important to underline that this visualization has been designed thinking to children and teenagers. So we are trying to call their attention and try to focus their attention. So the idea is to give them a different kind of content, so they can trying to calling their attention and trying to communicate the concept and the definition we want to give them. Okay, so in this case, our objectives are not scientists or researchers about this topic. So just to show you some more concept, for example, if we are interested in knowing why this research, this project is important, can be considered relevant, we can enter in the why bubble, and we can see, for example, the benefit of this project, like the economic benefit, the health benefit, and so on. If we want to explore the benefit, the health benefit, for example, here we can see like some of them, like sugar reduction, or innovation, or genetic disease, or nutritional quality, and so on. So if you are interested in trying this application, or also the other one, the other game that Paloma showed, you can contact with us and we can arrange a demo for you, or we can help you in using this for your purposes.
Okay, thank you, Teresa. So can we already play it when I have a VR helmet at home? Can I go to the link that you provided in the chat and then do the game myself?
Yes, you can try it.
Okay, thank you.
Consider that is a prototype, because we really don't work with final product. So we generally work with prototypes. So always there is some bug that you can discover. We try to solve all of them, but sometimes it happens that you could have some technical problem with it. So just let us know.
Okay, and the final version, the non-prototype, will be published on the website of CHIC?
Yeah. Now okay. I'm speaking because I will be responsible for it.
Yeah. Go ahead, Paloma.
Yeah. We are using a very novel technology for implementation, which is called Webvr. The main benefit it has is that it can be used in different platforms. So with the same codification, with the same program, Teresa is now using it in the computer, and if any of our participants link into the URL that Teresa shared in the chat, they can use it with that. But you can also use it with virtual reality glasses. So it's really portable to different platforms.
The only thing is also on my computer, a gaming console.
Yeah, you can use with the computer. The only thing you cannot use it yet, but we are creating code, is with tablets. It's because of the kind of interaction.
Oh, yeah.
This is thought to be used with the 2D handles that Teresa showed, okay? Which is very typical from virtual reality, because it gives better efficacy than using the hands or using any other kind of device. But if you use it in the computer with your keyboard, it will work. What we are doing now is implementing a version for touch screens, but it's completely different, because with touching you have to consider other kind of gestures, which can be done, but it's not exactly the same. But it can be used still in the computer. And the only thing, as I said, this is from the implementation point of view, is a programming language that is being developed. And something which is developed or is under development is being changing all the time. So we are doing adjustments, adjust several things whenever a new API is delivered. This is very technical, but in programming languages that are evolving, there are things that change from one version to the other. So when the version of the language changes, you have to do small modifications. And these are the bugs that Teresa was mentioning, that whenever this Webvr launches a new version, we have to change some small things in the code so it keeps working. But I invite all of you to try to use it. And as Teresa was saying, our goal with this kind of tool was to go to schools and to fairs. We were a couple of times in Aula, which is the biggest educational fair in Madrid. And there we could interact with young students. And we saw, as we knew, because we work in this, that these kind of technologies are very engaging. And it's a way to engage people. And once we are there, then you learn something that is also useful. And for us, it was very interesting because there is a lot of fuss and not really knowledge about gene editing and many preconceived ideas. And being able to work in an educational tool that can help to avoid this kind of preconceived ideas that are not right, or even we could say the fake news about gene editing that seemed to be something really bad. That was something that was really interesting from our point of view.
Okay, thank you. It looks exciting. I would like to wear the helmet and try to, at least the VR looks like
well,
a
it's
helmet.
It's a completely different experience, I think. In a computer, it's kind of boring. But when you wear the helmet, as I said before, since you are not anymore here, and you are there, and you have
feels
this
like
impression, that you come into the bubble and you get inside the bubble, this is something that has to be experienced.
Okay. Thank you, Teresa and Paloma.
Thank you very much.
So, gives us some time for questions from our attendees. I also saw a question from Abdelatif about sugar versus bitterness, and I think Matthew was able to answer that question. That's nice. Thank you, Matthew. Other questions for our computer department, gaming department? So, Paloma, what do you expect that we can show the game with the challenge? When can it be used at schools?
Yeah, I think that by the end of this year, it pretty much depends on the situation, but I hope that we will have a version. I'm not sure when we will be able to go to schools again.
Yeah.
Our last experience was on March 8th.
Oh, really?
Yeah, just
bad,
before
right?
the lockdown in Madrid. So it was quite complicated because we were cleaning the classes all the time, trying to, because we then knew that things were complicated. But if you are careful and you have a small group, the only thing you have to do, and we did a demo the other day for a company about a different product, but using also the glasses. What you have to do from one person to the other is just to clean very well the glasses, and this is another job yet.
Okay.
But I think we will be able to do it in schools. I hope so.
Okay. So I think I do not see any questions coming in. That leaves it up to me to close this online meeting of CHIC. I would like to thank all of the members of the panel. Teresa, Armin, Matthew, Paloma, Anna, and Katja, thank you for being with us. Our audience, thank you for being with us. Keep an eye on our website, on the CHIC website, when you want to know more about the progress being made on both the scientific part and on the gaming part. I wish you all the best for your health, for you and for your family. Stay safe and hope to meet you again.